do I advance the slides? This? Okay, sorry, everyone. All right, disclosures. I'm an inventor on pending patent applications for MRI methods to measure CSF flow. So it's really fun to give this talk here today. I think this might be one of the first times that there's a session specifically on cerebrospinal fluid, or CSF, here at OHBM. And I think this partly reflects that this is a really kind of rapidly growing and evolving field. There's starting to be several posters on this here at the conference, and I think that there's a lot that we can suddenly do as new techniques for MRI of CSF flow are emerging. So I'm going to be focusing on the cerebrospinal fluid, or CSF. Which is essentially this bath that envelops the brain, cushions it, and is really essential for the health and function of our brains. And CSF is often ignored, I think, in many imaging studies, but it's always there, and it's always dynamically moving and pulsing. And so it's a really kind of good system to understand and begin to be able to analyze and interpret. So today I'm going to cover 5 topics. It's sort of in the time that we have. Firstly, kind of motivating why you might want to image CSF flow. I'll give a brief introduction to CSF biology, just to kind of give background to those who may not know it, and then discuss different MRI techniques that can be used to image CSF flow in humans, what we know now about CSF dynamics, and then finally, even if you don't want to study CSF, how you might need to take account of these dynamics in fMRI studies. All right, so the CSF has really diverse biological functions. And the first is that it acts simply as a cushion. So CSF is around your brain. If your head moves and your brain moves around, CSF acts as a cushion. If someone develops a CSF leak, where CSF actually is beginning to be drained out of the skull, they will experience really intense headaches and might even have to be hospitalized, lying down at an angle with their head below their body to prevent the sagging of the brain down into the spine. CSF also contains lots of molecules like glucose and other signaling molecules, and so it's actually a way for signals to be passed within the brain. And then finally, as Meryl alluded to in her initial talk, a core role of CSF for the brain is waste removal. So CSF truly is the bath of the brain, and it's this liquid that actually is responsible for clearing out all of the waste products that our neurons generate during the day. So here's just kind of a cartoon of how this works. In most of our bodies, the lymphatic system is responsible for this. So molecules can move more freely between the blood and the tissue. And then waste products are drained by these lymphatic vessels. However, in the brain, due to the blood-brain barrier, it's much more difficult for things to pass between the blood and the tissue. And so instead, we have CSF surrounding our blood vessels, and the CSF can pass and be exchanged into the tissue and help accumulate waste, which is then drained out of various pathways out of the brain. And so this waste clearance process is really important, and we can see that if it goes wrong, this also is implicated in really diverse neurological disorders. Here's just an example of one. So when we have too much CSF accumulating in the brain, this is called hydrocephalus, and the most common form of this is idiopathic normal pressure hydrocephalus, which can often occur in aging. And so here's an example. You can see this person's ventricles are really expanded here. And there's a large volume of CSF in the brain. Idiopathic references the fact that we don't know why, this just happens. And unfortunately, these people experience problems with their gait, with incontinence, and this actually leads to dementia. But what's quite remarkable is if you can diagnose it, put a shunt in, and drain that CSF, you can actually sometimes reverse this before the dementia actually occurs. There's also been a really strong increase in waste clearance because of neurodegenerative disorders and especially Alzheimer's disease. So we know, for example, that Alzheimer's is linked to the accumulation of proteins in the brain, such as tau. And in this image, you can see that if you scan where tau is in someone's brain, it's particularly those regions where this protein is accumulating, where you also tend to get atrophy of these of the gray matter. And so it's thought that actually a breakdown in waste clearance leading to the accumulation of these proteins might be a critical factor in the development of neurodegenerative disorders. This is also just to say, since this is the brain-body session, one thing to keep in mind is that CSF is also a route through which the brain can actually communicate with the body, because the CSF contains these really diverse signaling molecules that are then actually transmitted through circulatory system, lymphatic system, and really pass into the rest of the body. So CSF serves all of these critical functions. It's implicated in really a wide range of different disorders, which I think each provides a reason why you might be interested in CSF itself. But even if you're not, and if you just do fMRI, CSF is in your data as well. And so here's an example of an fMRI acquisition where you can see here in the third ventricle and the fourth ventricle, these really clearly pulsing flow signals. Okay. And so these are present regardless of whether you're interested in CSF or not. And the nature of these dynamics are quite complex. And so it's not something that you can simply regress out without having lots of follow-on consequences to the rest of your data. All right. So how do these CSF systems work? So I'm going to start with just the production. So it's thought that most, probably about 80% of our CSF is produced by this organ called the choroid plexus, which is this heavily vascularized structure that's hiding here in the third ventricle and the fourth ventricle. And this secretes the CSF and fills it up in the ventricles in the subarachnoid space of the brain. And this process is really active. So we make as much CSF as our whole skull and spinal cord contain about 4 times a day. So 4 times a day, we're making a fully fresh new batch of CSF, and that full volume is also drained from the brain. So this bath is constantly as if the tap is always open and the drain is always open, and the fluid is constantly circulating. So as CSF is made, it flows through the ventricles and makes it into the subarachnoid space. And this is kind of the textbook view of how CSF circulates with these arrows, the idea being that it's made in the choroid plexus, it fills the ventricles, and also flows dynamically through the subarachnoid space. And this flow is always happening. But one thing that's important to remember is that these arrows that you'll see in the textbook version really reflect the net way that this flow is moving over many hours. If we actually look over just a couple seconds, the CSF flow is actually very dynamic. It's pulsing in waves back and forth. And so it's not just respecting the directionality of these arrows. In fact, it's as if there's kind of ongoing waves throughout the ventricles and subarachnoid space all of the time. Okay, so if this is a waste clearance system, it needs to not just be around the brain, it needs to actually also get into the brain. And so the way that this happens is through these perivascular spaces surrounding blood vessels. So our blood vessels will dive into brain tissue, like you see here, and around blood vessels is a perivascular space with CSF. The CSF, through much debated mechanisms, exchanges into the tissue where there's interstitial fluid, so that's the fluid kind of surrounding our cells, and eventually makes it out again, carrying with it waste products that need to be drained. And there's a couple different models for how this might occur. So a lot of work has been done elucidating this glymphatic system model, which you may have heard of. And so that says that as we move away from our conventional model where we're making CSF, it goes through the ventricles, it goes through the subarachnoid space, and then it makes it into the tissue. Through this model, it's transiting through this perivascular artery space, passes into the tissue, and then is drained out by the veins. And one thing you can immediately see as people who, many of you study neurovascular coupling, you look at neurovascular signals, is that if the blood vessels here change their diameter and get larger and smaller, that's going to directly affect the movement of the CSF in the space immediately surrounding it, right? Which is also going to be pumped as blood vessels change their mechanics. So under this model, eventually this makes it through into the tissue and takes waste products out through the veins. In alternate models, there's also this idea that maybe we don't just have directional flow, but it's really mixing, which could involve waves that don't require a specific direction of flow, but nevertheless are able to kind of clear waste, as well as the iPad model, which Meryl referenced earlier, which is essentially suggesting that maybe these waste products exit through the arteries instead of through the veins. And MRI is starting to become a tool which can actually explore these different models. So here, for example, they did MRI study of different tracers passing through the CSF systems and implemented computational models predicting where should this go depending on which of these models is true. I think providing really an opportunity for MR to start to inform the mechanisms of how these systems work. And in general, where our CSF goes is really a surprisingly active area of research with a lot of different possibilities. It's thought that it goes partly along veins, also passes along the cranial nerves, and also drained by the meningeal lymphatics. And just sort of in a testament to how new this field is, these meningeal lymphatics were only discovered in 2015, so less than 10 years ago, really demonstrating that we're still learning a lot about how this system works. So all that to say is that waste clearance is a process in the brain that takes place at multiple spatial scales, and we really need to be able to look across those scales to fully understand what's happening. CSF is being secreted and exchanged at really large volumes, about 500 milliliters per day. It moves through the ventricles and the subarachnoid space, but ultimately we also need to kind of have tracers in these spaces to understand where it's going. And then finally, it makes it around blood vessels and into the tissue and ultimately out of the brain. All right, so how might we study these processes with MRI? MRI is a really ideal tool to look at this because, in a lot of things that we study in the brain, rodent researchers or other sort of animal model system researchers may do quite well by opening up the skull, putting in electrodes, putting in optical fibers, and really invasively exploring the system. The problem is that if what you're studying is actually a fluid system, opening the skull has really dramatic consequences on our intracranial pressure and on where that fluid goes. And so this is a case where MRI is kind of a uniquely powerful tool for letting us look at the system without disturbing it and actually kind of tracking the dynamics that naturally occur in this endogenous system. And so you could imagine, just like we have functional and anatomical fMRI for looking at neuronal activity, we may want to do the same for considering CSF flow. And so I'll just briefly talk about some of the most common methods that kind of take the functional perspective of measuring the flow, the movement, and dynamic transport of fluids in the brain. And then also you can think about anatomical methods to study this. It could be that you just want to look at the size and morphology of the ventricles or the spaces that it's moving in, and in particular the perivascular spaces, which are right on the edge of what we can see with MR. All right, so the most common and standard tool for studying CSF flow, or really any fluid flow with MR, is phase contrast MRI, which is not so common in the world of fMRI, but is extremely common in the clinic. And this has been around since about the 80s. This is just an example of what this looks like. So you can see, you could imagine having a magnitude image, which is what we typically look at in fMRI, right? Where you can see the magnitude of the signals that you're looking at in the image. But we also have a phase image. And in phase contrast MRI, this phase image is used to actually extract velocity. And so this can be a very powerful method for looking at fluid flow because it reports velocity in quantitative interpretable units. So for example, in centimeters per second. So if you do phase contrast MR, you're able to actually say for a given slice, CSF was moving at exactly this speed at exactly this moment in time. When phase contrast was first developed, it was pretty slow, and so for the most part, for most of its existence, it's been used to look at cardiac-gated flow. So we need to average over something, and people would typically average over the cardiac cycle and see kind of fluctuations in CSF flow locked to the heartbeat. In the last few years, real-time phase contrast MRI methods have been developed, and so now this can be done at really high temporal resolution. And with that, people have started to notice much more complex dynamics in terms of how CSF flows. The challenges with this method are more that it has lower sensitivity to slow flows, and so if you try to look at kind of slower and slower flowing fluids, phase contrast may sometimes lack the sensitivity to give you a really good image of that. There's begun to be a lot of interest in using diffusion MR to look at waste clearance systems of the brain. And there's many different ways to kind of try to analyze these properties. So I'll just kind of comment on them at a high level. The main thing being looked at here is the idea that maybe we could actually look at that really microscale movement inside of the tissue, reflecting how water is moving at this very tiny scale. Here's an example of one type of analysis that's starting to become done, which is this DTI-ALPS index, with the idea being that if you could look at water diffusion in different directions and consider how much is diffusing along the perivascular space, perhaps that could give you some information about the way that solutes and fluids are being transported in the tissue. So diffusion MR is a really kind of exciting area because it does give you this microscale view into what's happening inside the tissue, which is still really not known. I think the challenge with this method is that it's more difficult to interpret exactly what you're measuring. So phase contrast, you could look at the CSF. You could say, I know that the CSF is moving at exactly 2 centimeters per second. In this case, there's actually probably many different changes to tissue properties that could change these diffusion values. And so I think it's important to consider, if you get a change in these metrics, what types of physiological changes could produce that, part of which might be changes in waste clearance and part of which could reflect really other things going on in the brain. A very kind of newly accessible way to start to look at CSF flow is by using fast fMRI. And so I put TR less than 500 milliseconds here, but that's not a rule. It's just kind of a guideline in the sense of when will you be able to see flow with the highest sensitivity. And so the idea here is if you are to do fMRI at very fast timescales, so with a TR of less than 500 milliseconds or so, you actually become sensitized to flow signals inside of your data. So this is still the same kind of sequence that you would use for fMRI. But as your imaging speeds up, the edges of the acquisition volume, so for example, this bottom slice here, and this top slice here, become sensitized to flow effects. And so you can see if I blow up the bottom slice, you have this really bright signal here in the ventricle, and that signal is actually reflecting the upwards flow of CSF. So why does this happen? So essentially, as we accelerate with fMRI, you might have noticed if you've tried doing your scans faster and faster, your images will get dimmer and dimmer. And part of what this is reflecting is that if you scan really quickly, right, your very first image will be bright, and then after that, the rest of them will be dim. And that's because if you're kind of tipping all of your protons over with your RF pulse and you allow them to come back up, if you're scanning rapidly, you tip them again before they come all the way back up. So you no longer have this much contrast from a 90-degree angle, you just have a little bit of contrast. So everything in your image is relatively dim. However, if a new fluid flows into your acquisition volume, which you've never imaged before, it's still going to be bright. It hasn't experienced any RF pulses yet. And so what this means is that the signal intensity at the edge slices of the fMRI volume will actually be conferred by the velocity of a fluid that's flowing. And so if you look at what this looks like here, if you had fluid that was flowing upwards really slowly, you would see a bright signal just in your bottom slice, and then it would decay away. And if you had a fluid that was flowing rapidly across multiple slices, you would see this flow signal in multiple slices. And so what this means is that the velocity of a flowing fluid is going to be reflected by the relative intensity of the fMRI signal in the edge slices of the volume. And so this is a although you can do this with the same sequence that you would use for standard fMRI, this is a really different contrast mechanism by which your signal is coming. It's not the bold response, it's actually a flow signal. So this is just an example of what your voxel time series could look like if you end up scanning rapidly and sensitizing to flow in this way. So I've pointed out with these orange arrows when you're getting large upwards flow events. And you can see, for example, here, there's this really strong signal in the second slice, which is smaller on the third and even smaller on the fourth slice. Whereas over here, the fluid is flowing upwards even more quickly, and we can actually get this bright signal across multiple slices. And so this means is that actually by looking at the intensity of the signal in the edge slices of fMRI, you can get a readout now of velocity. But very conveniently, it's still an fMRI protocol, and so you also still have hemodynamic signals at the same time. So strengths of this method are that you're able to simultaneously measure bold in the whole brain, as well as having a separate CSF flow measure specific to the ventricle. It's a very fast technique with good temporal resolution, and it has really high sensitivity to slow flows. A downside is that this is not a quantitative measure. So if you were doing phase contrast, you would be able to say 2 centimeters per second. Here, these are still in arbitrary units, and we're still working on translating these arbitrary units back to interpretable quantitative velocities. So if you would like to do this in your own data, how might you measure CSF flow with fast fMRI? So the shorter your TR is, the stronger this flow-related enhancement will be. So again, this isn't an actual cutoff. It's just that it's under about this range that we tend to see the brightest flow signals and be able to have the highest SNR if we're trying to look at flow. But a second thing that's really important to consider is that the flow signals are really generated by entry into the imaging volume. And so you're going to have the strongest flow signals at the entry slice, which means that you would need to position your acquisition volume in such a way that your edge slice intersects with the area that you'd like to measure the flow in. The other thing to consider is that you would want to position it in exactly the same way in every subject, because again, if you position it too low in subject A and up here in subject B, then you're actually going to be measuring flow from different positions and different regions in those two people. You can't go back later and measure flow in a region other than the one that you originally set up your acquisition volume to. And then the final thing to consider is that it's measuring flow inwards into the slice. So here in this case, for example, we would be measuring flow up. Up here, we would be measuring flow down. And so that's part of why it's easiest to measure this in a region that's relatively narrow, where you know what direction the flow is going in. If flow is very turbulent, you're going to have a harder time measuring it. Okay. And so just rounding out methods that can be used with MRI is this invasive strategy. So everything I've talked about so far is really relying on just movement of water in the brain. But if you're interested in waste clearance, you might actually want to say, well, how does a larger molecule transport in the brain? For example, if you cared about amyloid or tau, these are pretty big proteins that need to be transported in different ways. So very, very recently, some groups have begun conducting these intrathecal contrast agent studies. So for this, they'll actually inject an MR contrast agent through a lumbar puncture, right, through the spine into the CSF, and then image the movement of that tracer from the spine into the brain, into the tissue, and eventually out of the brain. And through doing this, they'll image at multiple time points over as much as 48 hours because it seems to have, a half-life of about 20 hours or so to make it out of the brain. To my knowledge, this is not currently done in the US, but there's several groups in Europe. And here's an example where you can see the movement of this contrast agent eventually into the tissue, lighting up here after 24 hours. Finally, you might be interested in the anatomy of what these spaces move through. And one thing that's really striking is that these perivascular spaces in the brain enlarge as we age. And the bigger and more numerous they are, as seen on MRI, the more likely someone is to be diagnosed with a neurodegenerative disorder. And so it's become of really high interest to measure these spaces since they seem to be consistently linked with onset of neurological disease. Although, of course, I should be very clear that they do also appear in the completely healthy brain with a load that just increases relative to age. And so this is an example of what these look like, these dark spots here. Again, this is not a clinical diagnosis. It's just a sort of scientific observation that in addition to increasing with age, particularly the number of where these are, is correlated with Parkinson's disease and Alzheimer's disease. And you can see that MR now provides a really clear way to visualize these spaces, which is relevant for studies of these disorders. Okay, so just to kind of show you now what could we do with these methods, I'd like to just really briefly give you a very quick overview of some of the studies that have looked at what CSF is actually doing in the brain. So we know that our CSF pulses with every heartbeat. So with each heartbeat, it's going up and down. This is a really fast phase contrast study that was performed here. And you can see, for example, in the aqueduct here, this pulsation of the black CSF signal with every heartbeat and also with every breath. So you see this smaller kind of filtered blue oscillation of the signal with the faster cardiac oscillation riding on top of it. And this reflects that our breathing also changes CSF flow, in part due to just pressure changes driven by the lungs. And so our CSF is constantly pulsing during wakefulness, locked to respiration and the cardiac cycle. There's also, however, been a lot of really interesting studies from animals looking at sleep, which have suggested that waste clearance actually seems to be much higher during sleep than it is during wakefulness, and sleep might be a particularly special state for transporting waste out of the brain. And so in our group, we've been using these fMRI-based CSF flow measurement techniques to look at sleep. This is an example of EEG if somebody's sleeping inside the scanner. So here they're asleep and here they're awake. And you can see that when this person is asleep, the CSF starts to pulse in these really huge low-frequency waves, about once every 20 to 30 seconds. And this is happening in a really kind of striking manner that's much larger than the waves we see during wakefulness, which here actually are just the respiratory cycle, just like I described to you earlier. And you can see these are really tightly locked to the vigilance state someone's in. So when this person wakes up, these large low-frequency waves of CSF are suppressed. So where are these coming from? What could be driving these really kind of strong waves of CSF flow? What's really striking is that if you use this fMRI method to measure CSF flow, you actually have the bold signal at the same time, so you can look at hemodynamics in the brain. And it turns out that these are really tightly anti-correlated with the CSF flow. And this is for the reason that Meryl mentioned in her talk, the Monroe-Kelly doctrine, which is that if your blood volume expands, there's nowhere for the CSF to go but out. If blood volume drops and vessels constrict, that leaves space, allowing CSF to flow upwards into the brain. And so this leads to this constant coupling between large-scale bold and CSF flow. And this is a video of someone sleeping inside the scanner just to give you a sense of what this looks like. As a reminder, we can measure bold, throughout the brain where we're imaging, so the red will be bold here throughout the cortex. The CSF flow we only measure at the entry slice, so I'm going to show that just here in the ventricle. So what you can see is that while this person is sleeping, hopefully it plays, you get this wave of bold and then CSF flow, and these are coupled. So bold, and then CSF. Bold, and then CSF. And while this person is sleeping, these alternating waves of blood and CSF continue to oscillate. What we noticed is that before each wave of CSF flow, about 6 seconds earlier, there was actually an increase in neuronal, a change in neuronal dynamics, specifically an increase in the slow wave activity in the EEG. And this led us to the idea that actually neuronal activity can be one of the modulators of CSF flow, in addition to the cardiac and respiratory dynamics mentioned earlier. So basically, the idea is that since changes in the vasculature and blood volume drive CSF flow, well, we in fact are all familiar with something that's really strongly driving changes in the vasculature, which is changes in neuronal activity. And so neural activity, by modulating blood vessels, we proposed could actually change the flow of CSF through the ventricles. In addition to neuronal activity, autonomic state has a really profound influence on the vasculature. And Pinar Osbe has done a bunch of really great work on this topic. She's going to talk next, so I'm not going to go into it. But essentially, both these autonomic and neuronal changes modulate the vasculature and thus can kind of drive the CSF flow, which means that the brain actually has active control over the fluid that surrounds it. And just to kind of causally test this idea, Stephanie Williams, a PhD student in our group, used an intense visual stimulus to drive a lot of bold response and neuronal activity while measuring CSF flow here in the ventricle. And she actually found that even this simple sensory task could really strongly drive CSF flow in the ventricle, where when the stimulus would go off, blood volume drops, and then CSF flow actually flows up into the brain. Really excitingly, this study from Xiaolu's group at Penn State also showed that the CSF flow dynamics could actually be detected in the ADNI dataset, which is a conventional fMRI dataset, demonstrating that, in fact, this signal is present in quite a lot of fMRI data. One way you can tell is that he was able to identify this consistent anticorrelation between the CSF signal in the ventricle and the global gray matter bold. And by doing this, he was able to show that specifically in patients with Alzheimer's disease and mild cognitive impairment, this blood-CSF coupling is impaired, suggesting that this indeed seems to be implicated in neurodegenerative disorders. So that's some of what we know about CSF. What don't we know? A lot. I think this is a really new and emerging area where there's a lot that's still to be discovered. We'd particularly like to understand this intersection between large and small-scale CSF flow and understand better the functional role of CSF and why it's implicated in so many disorders. And just to quickly wrap up, I'd like to talk about what does this mean if you're just doing fMRI and not looking at CSF flow. And as you can see, the CSF is always moving, in this case actually by quite a bit. And so the CSF signal in fMRI is often regressed out, and that's because of this first point, the concern that CSF signal is really representing bulk motion, which is definitely the case. However, even in a typical fMRI data set, there's often going to be smaller contributions to the signal from flow, which can actually also be related to blood volume and brain activity. So we know, for example, that bulk motion will really change your CSF signals, if only even because your voxels with CSF in them are going to be located in a different place. But also tissue motion is driving CSF signals. And in fact, some of this tissue motion is real kind of pulsatile activity that might be quite interesting to consider. There's also a series of excellent studies showing that when you have neuronal activity driven by a task, that induces volume changes that displace CSF. So this motion can be expected to occur from task studies. And then this really important study from Gonzalez-Castillo et al. A couple years ago used the HCP data set and found that they could actually detect flow-like signals even in the HCP data set where the TR is around 700 milliseconds, suggesting that the CSF signals are there and they're really strongly correlated with vigilance state, with much higher CSF flow signal in people who are in low vigilance. So I think something to just be aware of if you're going to regress out CSF signals is that the CSF signal has a lot of functional meaning to it. It co-varies with vigilance state, so the effect of doing this is going to depend on what vigilance state your participants were in. And it's actually correlated with large-scale neuronal activity, both because of local neurovascular coupling and also because neural activity changes with autonomic state. And so since CSF timing is delayed relative to neural activity, regressing it out can have complicated effects. So it might be for your task in your study, that's the right thing to do, but it might be that it's also introducing signals you might not have intended, and so I encourage you all to look at the CSF in your data and consider what it's doing in the task setting that you are using for your study. Okay, so to wrap up, CSF is really important for brain function. There's many open questions on how this works. And also a wealth of new MRI tools are just being developed to allow us to study CSF flow dynamics at fast time scales. These CSF dynamics are present and embedded in fMRI data sets and have really complex relationships to neural activity. And if you'd like to know more, I'd like to point you to this review article that came out of a workshop that we recently held. So this came out last year, and it gives a nice overview of kind of current state of using MR to study CSF flow. Thanks.
Thank you very much, Laura. We have time for one short question.
It's a fascinating talk. And my question, if we, earlier, if we understand the brain volume, including CSF vessels and tissue, is constant, then shouldn't we expect that the fMRI signal increase and the vessels dilate and CSF decrease to keep that constant volume? Therefore, there's a precise anti-correlation, so there shouldn't be any delay. But the movie shows that one thing goes and the other thing follows. And just wonder.
Yes, great question. So your intuition's totally correct, right? The blood and the CSF should be doing the opposite thing. But I think it's really important to keep in mind that the bold signal is not a blood volume nor a blood flow signal. And so that's, I think, a really kind of critical piece of this, which is that what we're measuring is related to and sort of sometimes used as a proxy for, but not actually a measure of flow or volume. One thing we often do is actually even take the temporal derivative of the bold signal, which is more closely linked to how flow is changing, and that already has a much better relationship to CSF. But in general, also the timing, you can see the CSF, it's moving at centimeters per second, right? So there's a literal physical delay of the fluid actually kind of making it in or out. So what that time delay is actually going to depend quite a bit on where in the CSF system you measure things, because something that's changing in the cortex might take a while for fluid to actually make its way from the ventricles. Yeah.
All right, thank you very much. We still have some questions, but we can do it in the panel discussion later.
I'll take them later.
Yes, thank you very much, Laura. Please join me in thanking.